Hey everyone, welcome back. Today we're going to do some notes on DNA structure and history. If you're wondering why that DNA is wearing pants and why those pants look a little familiar, it's because DNA has your genes. Haha. Anyway, so you might remember some of these things from when we talked about macromolecules, because DNA is a nucleic acid, one of the four types of macromolecules. But we'll start out with functions. So, three main jobs for DNA. One is to carry information from one generation to the next. Remember, that's genetic information. So, anything that makes an individual part of a particular species, as well as the information that makes it unique and related to its parents, all those changes are tracked in DNA. Next, determine the heritable characteristics of an organism. So those are all the physical traits, things that you can see, things that you can't see, that are all based on DNA. And then third is give every cell directions or instructions. And that's one of the reasons why we'll need to copy DNA or replicate it, which we'll talk about in another video. But every cell in your body is going to need a copy of this DNA because it's kind of running the show and it has all the instructions that the cell needs to perform its function. So, main components of the structure of DNA. So, remember, DNA is a nucleic acid. There are only two types of nucleic acid, DNA and RNA. For now, we're just talking about DNA, deoxyribonucleic acid. Because it's a macromolecule, it's a polymer. That means it's made up of repeated monomers over and over and over again. Couple things to add on to that. The monomers of DNA are called nucleotides. And on top of that, each nucleotide has three parts. So to make one nucleotide, you need three things. You need a phosphate, a sugar, and a base. All three of those things together make one nucleotide, and then a bunch of nucleotides together make up a DNA molecule or a polymer. So because DNA and RNA are both nucleic acids, they're both made of nucleotides. A couple things are the same, a couple things are different. So for RNA, you have a different sugar. Instead of deoxyribose, it's ribose, hence ribonucleic acid or RNA. The other difference for RNA is some of the bases. So some of the bases are the same, but RNA has one base that's different. But they both have nucleotides. Nucleotides have three parts for both of them. Hopefully this is review, but we'll cover it just in case it isn't. Four bases that you need to know for DNA: adenine, thymine, guanine, and cytosine. We call them A, T, G, and C. And the order of those A's, T's, C's, and G's makes you, makes you part of your species, Homo sapiens. It makes you unique and related to your parents, and all those differences and everything that makes you is tracked to a sequence in order of those four bases, those four letters, which is pretty cool. And then they line up in a specific way because DNA is double stranded. A is always across from T, G is always across from C. So what actually holds those two halves of a DNA molecule together? We'll see some pictures here in a second. There's a little sneak peek. But you've got two strings of nucleotides or two strands. They twist together to make this double helix or a twisted ladder kind of shape. And what holds them together in the middle is hydrogen bonds. Those are kind of the rungs of the ladder. And usually those are shown by dotted lines in diagrams. And what holds each string together is covalent bonds. Those are usually shown as solid lines because they're a little bit stronger than the hydrogen bonds in the middle. So here's the double helix, pretty familiar looking shape, but oftentimes when you see DNA drawn, you'll see it drawn flat like this because it's just easier to see what's going on and how things line up and what order they go in. So to get from this to this, all you're doing is you're taking the molecule and untwisting it and laying it flat on the paper. So most of our diagrams where we're labeling things are probably going to look like this, but remember that it does have a 3D shape like that. A couple things I want to point out. Notice how you can see individual nucleotides on this. So this DNA molecule, even though it's a bigger section, has these repeated units of a phosphate, a sugar, and a base. So we've got 4 nucleotides on the left, 4 nucleotides on the right. Next, look at the solid lines. Those are all covalent bonds. Oops. So these are all covalent bonds here, every solid line you see. So the things that hold this side of the ladder together are covalent bonds over here, hydrogen bonds in the middle. So between adenine and thymine, there are 2 hydrogen bonds. Between guanine and cytosine, there are 3. I'm not going to ask you about that detailed of an explanation, but as long as you know that there are hydrogen bonds in the middle, that's what holds the 2 strands together, or that's what connects the rungs of the ladder in the middle, you'll be good. Let's see. So some other things. Notice that the bases are in the middle. Those are like the rungs of the ladder. And then the sides of the ladder are alternating sugars and phosphate. Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. That makes the sugar phosphate backbone. That's this this blue and orange part that you can see on the side. And then the bases go in the middle to give us the 3D shape. Last little thing I'll mention, which is kind of a preview for replication. If you were to compare hydrogen bonds, the dotted lines, and covalent bonds, the solid lines, they're both strong. They're strong enough to hold this molecule together, but the hydrogen bonds are a little bit weaker than the covalent bonds. That means if we put stress or strain on this molecule, the thing that's going to break first is going to be the hydrogen bonds in the middle. So we'll see some enzymes when we get to replication that are going to twist and strain and stress this molecule. The first thing that's going to go is those hydrogen bonds, which will result in peeling those 2 strands apart, which is going to be important for 2 things, DNA replication, and then way down the road, protein synthesis as well. Okay, now that we got all that stuff out of the way, we get to do some story time. Yes, I love story time. This is a little bit of the story of how we got here, how we figured out all this stuff about DNA. Well, some of it at least. This is a really simplified explanation, but these are some of the biggest discoveries that happened to get us to this point. So the first one is an experiment done by Griffith in 1928. He worked with mice and bacteria. And the ultimate conclusion of this experiment was that something gets passed from one generation to the next. Some physical molecule gets passed from one generation to the next generation that includes information about that species or information about that individual. Today we call those genes. There is a video here if you want to watch it. It's kind of confusing. It's been confusing to people in the past, but if you want to watch it, open up the slides document and you can watch it on your own. So how did Griffith make that conclusion? So he dealt with 2 different types of bacteria in mice. One shown in blue here is the rough strain. Non-virulent means it doesn't cause the mouse to get sick and the mouse can live if injected with that strain of the virus. The next is the smooth strain, which is deadly. So if injected with the smooth strain, the mouse dies. So blue is safe, red is dangerous. Next was a heat-killed version of the smooth strain. So it was the dangerous bacteria, but it had been heated up, either treated with steam or boiled to the point where it killed the bacteria, and they were no longer dangerous at that point. That's one of the reasons why we cook our food. If you're eating meat, there probably is some kind of bacteria in there that could be harmful, but if you heat it up to a safe temperature for long enough, it'll kill them. So if they're dead, then the mouse lives and it doesn't get sick or anything like that. And then in the last column here, this is the one that actually let him conclude that there was something being transferred from one generation to the next, because look what he combined in this experiment. So he has blue, which is the rough strain, they're safe, and the heat-killed smooth, which are also safe, but together they're dangerous, and it results in death for the mouse. So why is that? Why would 2 safe things cause the mouse to die? And he concluded that something was getting transferred from the heat-killed smooth strain to the rough strain that actually made them transform into the rough, or into the smooth version, into the dangerous version. So what was happening, we know today, was there's DNA from these dead bacteria that got scooped up or transferred to the living ones that actually made them dangerous and made them deadly to the mouse. Griffith didn't know all those details, but he reasoned from the data that there was some information being transferred from one bacterium to another, some physical molecule that could transfer information. He didn't know that it was DNA yet, but he knew something was getting transferred. To kind of build on that work, 1944, Avery did an experiment to figure out what molecule was getting transferred from one generation to the next. So this is the experiment where they showed that DNA specifically is the genetic information. So what experiment did Avery do to show that that was the case? Using the same type of bacteria. So these are the heat-killed S cells, so the ones that were dangerous, but then they were killed. And then in the last step, you're going to see that we add the R cells, the rough ones. So those are the ones that were safe on their own, but they became dangerous when mixed with the heat-killed smooth cells. And what happened in between for the different trials is he added different enzymes to the mixture that would destroy one and only one type of molecule. So over here, he added proteinases. Those are enzymes that chop up proteins. So this sample had no proteins in it. And notice that it was still dangerous. The dangerous smooth cells still appeared at the end. Over here, ribonuclease, those are enzymes that chop up RNA. When you chop up the RNA, transformation still happens. The dangerous smooth cells still appear. And then in this sample over here, he added deoxyribonuclease, which is an enzyme that chops up DNA. Bing, bing, bing. And none of the dangerous cells appeared because the enzymes chopped up the DNA from the heat-killed S cells and didn't let that DNA transfer to the R cells, therefore not making them dangerous. So by going through the trial and error and adding enzymes to remove one component at a time, the only time that the transformation did not happen was when you got rid of the DNA. Therefore, DNA must be the molecule that transfers the genetic information to the next generation. So far, up to this point, we have evidence that DNA is the molecule that includes that hereditary information. Hershey and Chase did a similar experiment to try to reaffirm that or try to see whether it was DNA or protein. And they did it in a slightly different way using radioactive elements. I'll show you a diagram of it in a second, but first, this is a micrograph with color added to it so you can see what's going on. This big orange thing down here is a bacteria cell. And then these things here kind of look like little lunar lander type things or freaky spiders. Those are actually viruses. Viruses called bacteriophages that infect bacteria. These viruses are really simple. They have two parts, protein and DNA. So protein is everything you can see outside of the cell. And how they actually infect the cell is the protein part attaches to the cell and then injects DNA into it. So you can see the DNA entering the cell. So they used these viruses and bacteria during this experiment. So what they did is they did two trials. On the left here, they took normal bacteriophage viruses and they added radioactive sulfur to them that made them glow. And the thing about the sulfur was that's only found in the proteins. So they labeled the outside with radioactive sulfur, looked at where it lit up, looked at where the radioactivity went, and it stayed outside the cell. So when they made the proteins radioactive, they could see all the radioactivity stayed outside the bacteria, did not go into the cell. For the other trial, they used radioactive phosphorus, which if you remember cho, chon, chomp, phosphorus is found in nucleic acids, but not in proteins. So they made the DNA, or the nucleic acids, radioactive. And then when those bacteriophages infected the cell, they could watch the radioactivity actually enter the bacteria and the inside of the cell lit up. So that meant the part of the bacteriophage virus that was actually entering the cell that was causing it to change, get infected, change its genetic information was the DNA, not the proteins. So this confirmed that DNA was the genetic material. Okay, in the 1950s, Chargaff analyzed DNA, and he wasn't looking at the structure of DNA necessarily, but the composition. So he looked at the different rates of adenine, thymine, guanine, and cytosine in different samples. And when they analyzed the DNA of lots of different organisms, he saw this pattern that the amount of A was always really close to the amount of T. The amount of G was always really close to the amount of C. So he made the conclusion based on that. We call this Chargaff rule, Chargaff's rules today, or Chargaff's rule of base pairing, that says A always pairs with T, G always pairs with C. And this is, remember, DNA is so small, we can't just look at the molecule under a microscope. We can't see that kind of detail. But by analyzing it on a bigger scale, you could figure which bases have to pair up with the other ones. You might be wondering why these numbers aren't exactly the same. So, like, if the amount of A equals the amount of T, shouldn't the percentages be the same? What you're looking at is slight errors in the data. So, when analyzing the percentages, they were good, but they were not perfect. But you can see in every single case, they're roughly the same for A and T, roughly the same for G and C, which is how they made that conclusion. Last stop on our history tour is Rosalind Franklin. So this will touch on ideas that we saw in the Rosalind Franklin video from last week, which you definitely can rewatch. There's another video linked here if you want to learn more about Rosalind Franklin. But the long story short is Watson and Crick received the Nobel Prize, and they got the credit for discovering the structure of DNA. But Rosalind Franklin actually did a lot of the work. She took this photo called Photo 51 that was used to determine the structure, and she didn't get any credit. She didn't get the Nobel Prize. She died before she could receive recognition for the work that she'd done. And there's actually a rule with the Nobel Prize that you can't earn it after you're dead. So it's kind of too late to go back and to give her credit for the work that she did. But this picture was made using X-rays, doing something called X-ray crystallography, which is kind of like making shadow puppets with your hands, like you do like the bird or the whatever, different animals and things by making the shapes with your hands. It's kind of like if you can see the shadow, but you can't see the person's hands, and you have to figure out, okay, how would they be holding their hands in order to make this shadow? What shape would they have to make a shadow that looks like this? You can never actually see their hands, but you can see the shadow. It's kind of like that with DNA. And instead of using light, they're using X-rays. So if you take a sample of a crystal and you shoot X-rays at it and collect them, they're going to bounce off in different patterns based on how the crystal looks. Super complicated. I'm not going to expect you to understand why this shape means that DNA is a double helix. But there's just some cool stuff that chemists can do with X-rays. So in case you're curious about how that means DNA is a double helix, that's some really high level chemistry. But think about it kind of like shadow puppets. If you want to know more, check out X-ray crystallography. Pretty interesting field of chemistry. Okay, that is the end of these notes. If you have any questions, please let me know. Thanks for watching the video, and I hope to see you soon.